to your first. Hello all. Be able to see myself somewhere. Start my day. Can't connect to your webcam. What?
My WebEx says join meeting, so that looks fine. Looks like the automatic recording started.
Okay. Pause that. All right. Hello, everyone, and welcome to the 4-hour marathon that is Sage Arbor Foundations. So I am one of your 3 biochemists. Before we get started, Gardner, can you see the PowerPoint? And okay, great. All right. So we're going to spend a good amount of time, probably 10 minutes, just talking about some technology and how I do things. I do things a little differently than other faculty, so I will be speaking about just my lectures, okay? So while we're doing that, go ahead and try to take your first poll everywhere. So you can use an app, or you can go in a web browser to pollev.com/sagearbor. And all my poll everywheres will always be that same link, okay? So here I tend to use red for bad and green for good, which is known to be not so great because there's a significant percent of males that are colorblind in that. And so sometimes they use different shades. And so I try to get an idea of each year, how many colorblind people do we have? So in this emblem right here with the 2 circles, how many numbers do you see? And we have 14 results. So go ahead and I'll wait till that number gets up higher. We should have somewhere in the range of 160 students. Do you see the count of how many are logged in? Okay. And the chat box, let's bring that. Okay. And so while you're doing that, I will talk about a few things. So on my slides, I will have some emoticons that just kind of hint at things. So if you ever see a cutting board, that means that is very board testable, okay? So at the end of 2 years, you're gonna take your step 1 or complex 1. And so that basically means high yield material, definitely not a waste of time. You should definitely know this. Any sort of action figure guy or speeding means this is an actionable thing. So as a doctor, you can actually affect change by knowing this nugget of knowledge. If you see this memorized guy, that is, yes, this is something that you need to rote memorize. You won't see that often, but I try to make it very clear so you're not wondering the minutia. Usually, most of what we're teaching you to be is lifelong learners and to reason things out, but there's a significant amount of stuff of, yes, you need to know this vocabulary sort of thing. As I go I'll check at the chat box in particular when I have the poll everywhere and have a time to say something. So go ahead and you can type a Q in there if you have a question. You could type a C if you have a comment, or you could write a question, and I might not stop, but with 160 people, it's better not to just probably shout in. You can try that too. We're open to trying everything. But if you have a question, I'll address it or move on if there's a lot. If you see the FYI symbol, that means I am not testing you on this. So usually students are like, push it aside and don't have to worry about it. But sometimes it might connect dots more. It might be something I just find kind of interesting. I've taken a lot of these out because it was extra stuff the students didn't like. If you see an FYI with a hand, that means it's a hyperlinkable and you can click it. And if you see a clock in the FYI, that means it's an FYI for right now. I'm not going to test it, but later on, you will need to know it. So if you are, and this is again just my thoughts, but if you end up being kind of the over 80% student in your courses, then you can focus more time kind of learning things more in depth that you want to know, or studying for boards already. It's really not too early to do that. Okay, so we still only have 14 results. In the chat box, is anyone having problems doing the poll everywhere? I'm going to have poll everywheres throughout the lectures, so I want you to be at ease with them.
One person saying that.
Okay. Assuming that's one of the 14. All right, well, let's see. Of the 14, there was one colorblind person. So they found a number in the left circle. There is a reddish orange 12 in the right circle as well. Two people if they misclicked. Okay, I will try to point out if I have red or green things more. Small sample size, but useful. All right, I'm going to jump out of PowerPoint. Let me go a little bit further, and then I'll show you some. Okay, you're still in the PowerPoint, correct, Gardner? Okay, so in the top right corner of my slides, you'll see a version number. Those shouldn't really matter. The big take home there is sometimes I'll fix a typo or add something interesting, but that's not changing anything testable. And the version number would increment on the right. So if I had changed this last night or 2 nights ago, it could be version 3.03. That basically means you probably don't need to download it. If the first number ever changes, like to version 4, then I added an objective or I added something testable and you should re-download it. I'll mention if I ever do that, and I probably won't. Okay, so some links that will be kind of important. There's 3 big rules for the Sage Arbor lectures. Number 1, everything that is testable will be in the PowerPoint from the cover slide to where it says end of slides, okay? If there are slides after that, that is supplemental information. And if there's information on the sides outside of the slide, that's like extra stuff that I ended up not using, okay? So if you listen to my lectures, that's what you need to know, okay? The second most important rule is the same rule. Everything is in the PowerPoints. This is something students worry about, okay? So I'm about to show you a lot more than that, and that is all supplemental, okay? It's all if it helps you. So if you go to marian.edu/sage, that will go to my research page. So I'm going to show you that now. Yeah. Okay, can you see that? Pardon? Yes, okay. So the research page is not important, but at the top, there's a hyperlink list, okay? And if you click on that, it opens up this page. Now here you will see all my lectures, and then you will see a click to a Google Live document that is the lecture notes for that lecture, okay? And they're color-coded if that lecture note is for more than one lecture. So for example, this one is color-coded red because it's for the pH lecture, for the water lecture, and for the cystic fibrosis lecture. So you'll see that red again down here because in the spring, we'll have my cystic fibrosis lecture. So this will be a large Google Doc, but only a third of it is for the pH lecture. So now I'll go to that. Are there any questions about, hold on 1 second. Now I'm going to that document, which some of you are in. If you're logged in to Chrome or to Google, I'll see you, otherwise you'll be anonymous. Everyone has comment permissions here. So if you see typos, you can fix them. If you have a question, you can put it in, although I don't actively maintain this, but I do get emails. But if you do, what usually happens if you by mistake hit spaces or move things around, I see all that and I have to accept or reject it. So I wanted to leave comments. See, so for example, do I want to add a header? No. Okay. And I don't mind that, but it's more for a collaborative way for us to interact even though we're virtual. Okay, so the table of contents, this is by far the most useful way to use this if you want to. You don't have to use this entire thing. The table of contents is the first highest level of a study aid. And so basically it's all hyperlinkable, but it's an outline of my lecture. So since this is for all 3 lectures, it's pH, water, and CF, let's go down to the pH section. So we're going to talk about pH scale, DKA, diabetic ketoacidosis. Whoa, someone's doing a lot. Blood, pH and cellular compartments. And then if you can just read through this after the lecture and say, did I know what he was talking about here? Yeah, 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 yeah. And then if you don't, you can click on it and it will jump down and give you more detail often than in my lecture. Okay. Sometimes it's just a copy of my lecture, but a lot of times it might have much more detail. It might have a paste of a review article that goes into detail. You don't need to know anything extra. It's just to try to help you. Are there any questions about what I've said so far? Into the chat box. Okay. The last very weird thing I think that I do, there's probably more, but the last one that I can think of, oh, also, where can I get to that? I need to get back to that, hold on. Okay. In my, what was I trying to show you? Right, in that list of lecture notes, you'll also see a link. A lot of my PowerPoints are in Google Slide format. These are pretty much the identical one that I'm giving you. You can use them if you're not, but they might not be exactly the same. Okay, so I'd stick with your PowerPoint, but if it's an ease of use, I just have it out there online for free for the world. Okay, so again, everything you need to know is like everyone else in the PowerPoint in this recording. So let's keep going on now. And here are my objectives. So if you want to see my objectives like every other faculty member, just look at the objective column, okay? The weird thing that I do is I tell you how many slides have that objective to kind of give you a rank of the importance, and it's heat mapped here. So for example, describe the function of a buffer and biological system is on 7 slides, okay? And in addition, if you copy this unique code, they all start with OJV because that's not in any word. If you copy and search for that, control find it, it will jump each slide that has that is labeled by that. So as you study for the exam, if you want to study that objective, you can just jump through the slides that way. In addition, once this video is uploaded to Panopto, Panopto is searchable by any word. So you can search the video content by these objective slides. Okay, I believe that is takes care of all the Sage Arbor shenanigans. The objectives I try to make pretty granular, and I normally won't read through them. I'll read through a few to give you an example. So compare a compound's ability to cross biological membrane in its charged versus uncharged states. Assume no transport mechanism. So we're going to talk about aspirin as an example for that. The one odd slide, whoops, and I have them by topic, right, is for the cystic fibrosis objective. You'll get this whole objective again later in the spring. So I'm talking about some of it now, and I've crossed out the parts that you don't need to know now. So for the first exam, you just need to describe the effects and therapeutics of CF patients have for the pancreas, intestine, and lungs, just those 3. Okay, and so sometimes I purposefully am redundant in that this is amazingly highly board testable, and I want to give it to you once, test it, and then I might give it to you later, test it again. So it's a twofer if you learned it the first time. Any questions before I move on? How many students are on?
One question.
Dr. Arbor?
Yes, go ahead.
So when you post the recordings to Panopto, will you be posting them at the end of every hour block or at the end of the full 4 hours?
Probably at the end of the full 4 hours, I would guess. In
Okay,
the
thank you.
future, we're kind of working out bugs. What I'm doing right now is I'm actually recording the Panopto right now. And so in theory, it would automatically post, but I didn't have access to the folder it needs to be in. So I'm recording it somewhere else and I'll have to move it. But in the future for mine and others, if we do it this way, it should automatically go there.
We're trying to do it in 24 hours.
Yeah, so within 24 hours, and that will be the accepted, for foundations, the accepted spot of the video recording.
Okay, thank you.
Yeah, good question.
There was another objective.
The objectives will always be right after the cover slide. Are you doing the unified document again or no?
Yeah.
Okay, and in addition, we will have weekly Word docs that aggregate all the objectives for all the lectures for that week. But in all the PowerPoints, for example, here is my objective table, right? So there's 9 objectives for pH. Some of them are super small, right? Like one slide is list the pH of cellular compartments. Here's the ones that cover the water. This PowerPoint is 2 lectures in one, right? And then the lecture notes that are online also have the objectives. If those differ from the PowerPoint at all, they won't mostly. The PowerPoint's the authoritative source. Those might have had a nitty-gritty objective I decided to drop and forgot to take there. And if you see that, you can delete it and I'll see the note and update it. Good question. All right, so let's move on from there. I'm going to try to go like a few minutes long on the first lecture because I don't know if we'll have problems starting the other recordings before the scheduled time, so that would work out well. But we'll try to, I might go long and short. When I have large blocks, I like to kind of just take up what time I need in each section. So here's a case that you won't be able to do, but I want you to think about what you would have to know, and we'll start to chip away at that during this lecture, okay? So a 45-year-old female with a 32-year history of type 1 diabetes is brought to the ED, emergency department, in a state of confusion after a motor vehicle collision. Testing reveals a high fasting blood glucose, 650 milligrams per deciliter, and then we'll in the exam we'll give brackets like this afterwards that show the normal range. So normally you're 70 to 100 milligrams per deciliter. They also have a blood pH of 7.15, and urine is positive for glucose and ketones. Okay, so don't freak out if you're like, what the heck does this mean? Because that's the point. You are becoming a doctor. It will make sense. So what do you need to know for this sort of thing? What's the normal pH of the blood? I gave you the pH of their blood. What is type 1 diabetes? What does a decrease in insulin do? We'll talk about that with diabetes. And what does the increase in glucose in blood do? And so the most likely diagnosis for this is DKA, diabetic ketoacidosis. I'll try to leave things that are hyperlinkable blue here. Obviously, you do not need to click on this and know that now. This is very board testable and you'll learn it later, but the number one problem students have, is delving way too far into the weeds. My strong suggestion, definitely for my lecture and probably for many people's lectures, is to do a once over of the objectives. Kind of go through and be like, do I get it? Do I get it? And then you can kind of flag ones that you're not sure about and then go back. But you should feel good kind of running through and be like, okay, 70% of those I got or whatever. Okay, so let's talk about pH. Here on the right is a graph showing the pressure of oxygen on the x-axis and then the percent saturation on the y-axis of hemoglobin, HB, okay? And so we're going to use this. You've had this in undergrad. You'll see it a bunch of times here. It's very important. It's a good poster child of proteins, what they do, and it's important. So at a high pressure of oxygen, when you take a breath and your lungs have a lot of oxygen, the hemoglobin is almost completely bound with oxygen, right? And it doesn't really matter that much what the pH is, right? So at 6.8, a lower pH, 7.2, 7.6, they're all 95% or above, right? The hemoglobin all has a lot of oxygen. As you go down into the tissue range, okay, so as your blood gets pumped around and you're distal from your lungs in your extremities, and kind of coming back, you're not going to have as much oxygen. And then the percent saturation of hemoglobin drops. And we'll talk about this, and this is what you'd want, right? You want to dump the oxygen there where it's needed. What I'm showing you here is that the binding to hemoglobin changes more in the tissues based on pH. And so when you think, you kind of, a lot of times I might anthropomorphize enzymes. That means kind of like put human emotion on things that don't have neural nets to think. But so like, what does the hemoglobin want? And it wants to dump oxygen where it's needed, right? So in the tissues, I skipped slide 6. I'll come back in a second. I see that note. If you need oxygen in the tissues, let's say you're exercising a lot, and exercise, you probably heard, creates lactic acid. Well, acid is going to lower the pH. So if you exercise a lot and you lower the pH to 6.8, the binding of hemoglobin drops, which is exactly what you'd want because you need oxygen to be dumped there. Okay, so this has evolved to work the way our body needs it. And then drug absorption is often pH dependent. We're going to use aspirin as an example. Let me see if slide 6. Yes, I skipped slide 6. Thank you very much. So what is the pH? This should all be a review from undergrad, but you really need to grasp that it's on a logarithmic scale. So it changes tenfold for every 1 that you go up and down. So if you're talking about pH 7, okay, on the x-axis here is pH 7. If you go down to pH 6, that means that there's a tenfold increase in the number of hydrogen ions, right? A low pH means acid, and it means there's a lot of hydrogen ions floating around, okay? If you went from 7 to 8, there would be a tenfold decrease in hydrogen ions floating around, okay? And if you went from 7 to 9, there would be a 100-fold decrease in the number of hydrogen ions floating around. Proteins have evolved to kind of behave as they should in their compartments. So pepsin, for example, its peak reaction velocity, or where it's most active, which is on the y-axis, is in a very acidic environment, peaking here at around 2. That's what the stomach is. The trypsin protein is in the duodenum, and it peaks around 6, where which is the pH goes back up from the stomach to the duodenum. So that fits there. And then alkaline phosphatase, which is in the mitochondria, peaks at around 9, and the mitochondria, as you'll see in another slide, has that higher pH. I hear a TV, someone talking in the background. Are we all muted? I don't hear that. But if you have the TV on in the background, you got caught and mute yourself. Okay. See, I don't think I missed any other. Oh, a lot of people hear it. Okay. And I didn't miss anything else. Okay. So here is the first memorized slide guide. Okay. And so here are the different compartments in the cell and their pHs. So I drew this slide in a way that you can follow to go down in pH. So if you start at the nucleus, its pH is 7.2. Then when you go to the rough or smooth ER, you have a lower pH at 7. Then as you go to the Golgi has cis, medial, and trans compartments, and the pH continues to drop from 6.7 to 6.3 to 6. And then you go to the lysosome in my drawing, and it's acidic at less than 5.5, right? It's breaking things up. The mitochondria, I guess I should have started the mitochondria and gone into it's alkaline. So alkaline is the opposite of acidic, right? So it has a high pH at 8.0. And the cytosol is like the nucleus at 7.2. Okay, and the general trend of why and when I require you to memorize things basically is if you need your own boards, but also is this a small enough amount of information for you to retain, and is it eternal? Is it not going to change, right? The pH of these compartments is not going to change. It is central to the mechanisms that are going on in the proteins, and it's not that much, right? As a general trend. Now, I'm not going to ask you to differentiate, the 0.2 between the ER and the cytosol, but you should get large differences, right? The mitochondria versus lysosome versus Golgi, for example. Shut the chat. Okay. All right, so here we are with aspirin. I am never going to require for you to know a chemical structure. If I have it there, it's just to help you kind of understand what's going on, because we're talking about ions moving, and it's just black magic, I think, if you can't kind of think of it that way. So here it is, and we're going to talk about, is it charged or uncharged, okay? Now you can figure this out specifically, like how charged it is, what percent of it is, using the Hendel-Hasselbalch equation here. I'm not going to require you to do that. I want you to learn to approximate it, because that's good enough. And so as you swallow the aspirin, it's going to go into the stomach and into the duodenum. And so you have to know the pH. So everyone knows the pH of the stomach is acidic. And then later on, the duodenum raises that pH back up. Okay. So let me, someone type a C if they can comment and answer this question, and then it's like I'm calling on you as a raised hand, okay? So the aspirin ratio that is associated versus dissociated. Associated means this hydrogen is associated to the compound. Dissociated means the hydrogen has come off, okay? And I'm telling you that the pKa of aspirin is 3.5. The pKa means the pH at which half of it is associated and half of it is dissociated. Every compound that can take on and off hydrogens has a pKa for that hydrogen, and the pKa is where it's 50-50, okay? Now, at pH 1.5, will the aspirin ratio of associated to dissociated be higher or lower than 1:1? At 3.5, it is 1:1. Go ahead and type a C if you want to answer. And I, in class, wait for in silences, so great. Okay, Winston, unmute and let me hear what you think.
At pH 1.5, it should be 1 dissociated to 100 associated still.
Right, so very good. And can you explain why you thought that?
Yeah, so I mean, I did the Henderson-Hasselbalch equation, but
we got a gunner. Okay, you don't need to do that, but so the take home there is at an acidic environment. Remember, I said a lot of hydrogens are floating around, right? So this oxygen is negative and it wants to bind a positive hydrogen. And at the pK of 3.5, just think about, like, you can, get nitty gritty and talk about its energy landscape, but basically it has the energy to bind half of the aspirin molecules with a hydrogen. As you go down to 1.5, there's 100 times as many hydrogens. And so it's kind of getting rammed with hydrogen. So there's going to be a lot of them. Okay. So at the pH of 3.5, this ratio would be 1:1. Okay. Associated to dissociated, then it would be 10:1 and then 100:1. Okay. Oops, and I already answered that. Okay. So in the stomach, every time I have this box, I'm telling you, I'm quizzing you, but then you can just delete the box and see the answer. So in the stomach, we talked about it already having a low pH around, 2.5. And so we already worked through this, that it would be almost all associated, okay? Now, I want another C here for someone else that can comment. How is the speed of absorption affected by taking the aspirin? 1, on an empty stomach with food, 2, with calcium carbonate, and 3, with an enteric-coated aspirin, so aspirin coated with carbohydrate. And in the first, we'll do on an empty stomach with food, first food. Who has a C for that? Okay, Solomon. This is kind of one of the harder one of the three. Go ahead and unmute. How do you think it's going to
So I'm
change?
thinking on an empty stomach, the pH of the stomach is still lower, so it should be more associated. With food, maybe your stomach will produce more acid, so probably a little bit more still associated.
Right. So it's very true that it will be more associated. How will the speed of absorption be affected? So will the aspirin get into your bloodstream faster if you eat it with food or without food?
It should be faster
And why do you say that?
because there's no charge.
Why did the food change the charge?
Why does the food change the charge?
Yeah,
Well,
like let's say I have a headache. I could either swallow the aspirin with nothing. So I just had the aspirin on day 1. Then the next day I wake up and I have a headache and I swallow the aspirin with my lunch. Which day 1 or day 2, would the aspirin be absorbed faster? Sometimes this is one of those questions I'm asking. I kind of don't expect you to get it, but I want to hear reasoning out.
Well, based on my first reasoning saying that the food will produce more acid, so if there's more acid and the pH is decreased, then
Okay, I missed that the food would create more acid. So pretty much correct in that when you consume food, your stomach shuts to hold the food in there. So you're going to hold the aspirin in the stomach longer because of the food. And so the stomach's always pretty much acidic. And so this aspirin will be in the acidic environment more, and it will be associated. And the big thing that we'll be talking about here is when a molecule is uncharged, if there's not a transport mechanism like a protein bringing it in, it's easier to pass the a lipid membrane, okay, and to get absorbed into the bloodstream or go into a cell. And so you want an uncharged molecule to get through a lipid membrane, assuming there's no active transport. And for aspirin, there's not. We're talking about there not being. Okay, so good job. Now, does anyone want to answer the next two or one? What about with calcium carbonate? And if you put a C, maybe you can also tell us what calcium carbonate is. There's a common name for that. All right, Jose.
So calcium carbonate, it would decrease the rate of absorption because you become more charged. So following the same pathway.
Pause real quick, Jose. For everyone else, what is calcium carbonate? Do you know the common name? Like,
is that what Tums is?
Everyone uses the name brand. Yeah, we're not allowed to test with the name brand. It is Tums. It's an antacid, right? So go ahead with your answer now.
Yeah, so it would make the environment more basic, which would mean that less of the hydrogen atom or less of the aspirin or more would be dissociated. And so it would be more charged. It'd have more of a negative charge. So it would then not be able to transport as fast.
Right. Excellent. So day one, you already know if you go home and dad has stomach pain and he's taking antacid and an aspirin to dull the pain, you can be like, whoa, let's do those separately. Okay. Okay. What about enteric-coated aspirin? Someone else with a C. Or Jose, you can talk if you're still up. Someone else put a C then. Jose's out. Paul.
I'm thinking that it's not going to be able to be absorbed as quickly for the same reason as the previous one. The charge is it's going to be more basic, and so it's going to be less charged.
Okay, good thought.
More charged.
So an enteric-coated aspirin is basically aspirin with this carbohydrate around it. And the reason it was designed was aspirin can cause stomach bleeding. And so the idea was let's not have the aspirin in the stomach, and it can get past the stomach into the duodenum where the carbohydrates then digested, and then you have aspirin available. So the key there is that you're getting past the stomach to the duodenum. And in the duodenum, the pH goes back up. So that is not as good a spot for the aspirin to be absorbed because it's going to be more dissociated, more charged. Long story short, it still gets absorbed, but and another important thing to know is that the aspirin, it doesn't actually work directly on the stomach. So even if you have an enteric-coated aspirin, it gets into the bloodstream. It can still cause stomach problems. So I don't know a good scientific reason to actually buy enteric-coated aspirin. And if you went home and dad wanted relief quickly and was taking enteric-coated aspirin, you could now suggest that he just take the cheaper normal aspirin. All right, very good. Let's keep going. One more question. Can you please explain the empty stomach part briefly again? Sure. See if I can go up. So if you have, if on day one, I have a headache and I have a pain, back pain, I take aspirin with nothing. Okay. It's going to go in here, but my stomach's not going to close because that closes based on kind of consuming food or a certain volume. Right. And so the aspirin would dissociate some here in the acid environment, get absorbed a little, but it would also pass into the duodenum. And so in the duodenum, the pH was much higher. Okay, it went back up towards 6.5. And so there, the aspirin is going to be largely dissociated. Remember, its pK is at 3.5. So if you go up to 6.5, that's 4.5, 5.5, 6.5, 10, 100, 1,000 to 1 dissociated. So it's mostly dissociated and charged. And that makes it hard to get into the bloodstream because it has to pass lipid membranes. Lipids are hydrophobic and they don't like charges. We'll talk about this in the water lecture next. If on day 2, I'm like, that didn't work, and now I've listened to Sage Arbor's podcast, I'm going to take my aspirin with some food. When you eat the Big Mac, it shuts down it shuts the stomach here so the food stays in the acid to be digested. The aspirin also stays, so it's in the acidic environment, which is associated, which can be absorbed better. Does that make sense? Yes, that is why many drugs say take with food, right? Because of the charge of the drug, being less charged in the acidic stomach environment. All right. Okay, so what percent of the aspirin, I think I just went through this, in the duodenum, the pH is 6.5, and so it ended up being 0.1% associated because you went up 3 orders of magnitude. Now, what if the pH was 5, right, and the pK is 3.5? So far, I went up normal increments of 1. Again, you could use the Hender-Hasselbalch equation, but you don't really have to. You can just kind of intuit what it is to get close enough. So as you went up from 3.5, 4.5 would be 1 to 10, and then 5.5 would be 1 to 100, okay, associated to dissociated. It's mostly dissociated because you have less hydrogens around. But I overshot it. I went to 5.5. So it's somewhere in between 1 to 10 and 1 to 100, okay? Now, you could say it's 1 to 50. That would be wrong because it's going up kind of exponentially because it's a log scale. So you could also intuit that when you're halfway in a log scale, right, from 4.5 to 5, when you're at pH 5, is it going to be less than 1 to 50 or more than 1 to 50 associated to dissociated? Someone type a C and I'll let you answer. I'm going to try to annotate for the first time. Someone have a C for that? You got a 50-50 chance. I don't give a lot of 50-50. Well, yeah, I do, but. Okay, Benjamin, you're up on to answer, but before that, let me point out that I'll always wait for an answer. And if you guys take a long time to answer, it sucks up time, and then I have to say everything else faster. In addition, if I don't get through stuff, then I might just have like assigned reading. I used to have a lot of assigned reading, and I've taken it all out so I can just say it all to you. But if you slow me down, then that might come back. So let's keep it being as interactive as we can in COVID. Okay, you're up, Edwin.
Yeah.
I assume you want that?
Got shot in the dark, but I'd say be a little less than 50.
Very good. And why do you say that?
Because it's on a logarithmic scale. So as it gets bigger, it's going to grow more because it's a logarithmic scale.
That's right. So if
the rate of increase.
I hit the mic, dang it. If I was drawing a logarithmic scale, forget this curly thing. It would go like this if I was talking about, which way did I do this? Well, the way I drew it was dissociated to associated, right? It's going up as the pH goes up. And so if I did, let's say this is 3.5, 4.5, I'll use that one, 5.5, right? You're halfway here, but it's going up more later on. Okay, I don't know if that confused people or not, but basically you're right, like 1 to 30 would be a good guess, something like that. And now let's see if this randomly disappears when I go to the next page. Nice. Great. Okay. So let's talk about lactic acidosis. Here's an example of kind of something more complex that I'm just going to give you the high level of to talk about. So when you have ethanol, when you consume a beer or bourbon or whatever, wine, it can result in acidosis. And that's, let me get this annotate back. And that's partly due to the metabolic production of large amounts of lactic acid in the liver. So I'm not going to go through the details, but when you consume ethanol, it eventually becomes acetate. I'm just going to write ACT, okay? And as that happens, the double arrow means I skipped steps, okay? All of that creates NADH. Okay. Sorry for the clicks. We don't have a pen yet. We might work on that. Over here, I have sugar. So beer might have both of those, or wine. And that gets converted to pyruvate. I'm going to have a cleaner version of this for you to see in a second. Pyruvate. And pyruvate gets converted to lactate. Click, 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 click. The take home here is that in this process, from pyruvate to lactate, you use NADH to NAD+, okay? So you will hear me say this weird word that you've probably remembered from long ago called Le Chatelier's principle. Don't need to know that word, but what it means is that if you increase the reactant on one side of an equation, it pushes it to the other side, to the product. So in consuming this ethanol, you created NADH. So you increase the reactant here and you're driving over to more lactate, okay? So does it make sense to say that there's a buildup of lactic acid in the blood? We'll talk about that, I think, on the next slide, or is it? Okay, so estimate the ratio. Is this one working? Yep. Estimate the ratio of lactic acid to lactate in the blood at pH 7.3. Go ahead and type this into poll everywhere.
You remind me what
The pK of 3.65. Did I go back a slide? Dang it. 85? Okay. 3.85. Is this, and tell me if this poll everywhere is not working. For some reason I can't see how many, if anyone has clicked. And so poll everywhere, you can answer a bunch of ways. Like I know you can do it by text and stuff. There's, whenever I do a visual one like this where you're clicking on the line, it has to be in the browser. I assume you guys are doing it mostly in the browser anyway. I'm going to give you 5 seconds, and I would just keep a poll everywhere tab open. Not working. Okay. Well, okay. So I showed you that the pKa was lower, and so the pH is higher, right? So there's not as many hydrogens around, so it's going to be dissociated. And so this is one of the well, no, it worked. It worked for other people. Okay. Whoa, all over the place. So generally, you're more right. It's up here. Okay. I want the big trend, and don't worry that you got this wrong, because this was a learning poll everywhere. That was the whole point. People often misunderstand what an acid is, right? Because when you think acid, you think hydrogen, right? But lactic acid has the hydrogen because it has the hydrogen to give up. Lactate has already lost the hydrogen. Okay, so I think that usually students just take 1 or 2 times learning that to kind of get those questions right. Okay, so an acid has the hydrogen still to give up. Okay.
So for this one, would you view lactic acid as 4 and blood as 7 and just do that?
Yeah.
And be good enough?
Yes, the question was, how are you approximating that? For my level, you can just, as Dr. Gardner was saying, you could view the pK of lactic acid as around 4 and blood is 7, and you're off by 5, 6, 7 10, 100,000, right? So it's shifted drastically in one direction. And we'll get into buffers and talk about kind of the minutia of when you're off by 1 pH from a pKa, what does that mean, right? But if you're off by 1,000, it's drastically in that direction. Okay. Another question. Okay, Kristen, go ahead.
So just to clarify here, is it correct to say that when the environment's more acidic, then the molecule's more associated?
Yes, because there's more, if you're talking about the hydrogen, because there's more hydrogens around. So it depends on the chemical moiety that you're talking about. Like you might be talking about a pKa that's like an NH3 plus. So I don't want you to get confused that the associated is always uncharged. There's cases where you could add another hydrogen and that would make it charged. But oftentimes you're talking about like an OH where the associated version is not charged. Okay.
Okay, thank you.
Yeah. Okay. So I think I'm going a little slow, so I'll pick it up because I've already said all this. So in the blood, the pKa here is 3.85, and I'm showing you the structures which you don't need to know, and just so you can understand it. And so you see that it is basically you're increasing lactate. And this also goes for exercise, that when you exercise, people talk about having lactic acid. Well, the lactic acid kind of gave off its hydrogen and you have increased lactate, okay? 99.9% lactate in this case. Okay. Let's move on to buffers. So the general definition of buffers is that they can resist changes in pH. And so the long and short of it is that a compound is a good buffer when it is near its pKa, because at its pKa, it is 50% associated and dissociated. And so it can modify the buffer in either direction, okay? So it always has that dual capacity. Obviously, we have to keep certain compartments at a constant pH. So blood has buffers. These are very important biologically. Amino acids can act as buffers. We're going to go through the big ones. So here on the left is a generic amino acid, right? So N, C alpha, C carbonyl. That's the backbone. And then this R group is some side chain. If this was alanine, this would be a methyl group, a CH3. And if it was glycine, it would be a hydrogen, but usually it's some carbon chain. Since every amino acid shares this backbone that I highlighted, if you're talking about a single amino acid, it has two ends that have different pKa's. And so over here, we're showing the graph of the pKa's, but in short, the NH3 side, it can become NH2 or NH3 plus, and its pKa is 9.8. So at a pH of 9.8, half of this molecule will have NH3 plus and half will have NH2. The carbonyl side, the COOH, has a different pKa of 2.1. So at a pH of 2.1, half of this molecule would be COOH and half of it would be COO negative charge, okay? Now over here, we're showing that in a titration curve. So in the x-axis is the pH, and in the y-axis, we're titrating in base. So let's say we're talking about alanine, okay? So this R group is a methyl group, so it doesn't change. We start off at a very, very acidic, 1. And as we start titrating in base, okay, the pH goes up. And then when we hit pH 2, this COOH, down here, it was almost all COOH because there was a lot of hydrogen, right? As we go up at the pKa of 2.1, half of it has given off its hydrogen. So as you're titrating in base, this hydrogen is coming off and binding the base, okay? So that's why the slope increases. You need to titrate in more base to change the pH a little bit because the hydrogen is binding to the base, okay? Then you pass it, so all the hydrogen has come off, right? At pH 3, it's pretty much all gone. And then you can add just a very little base and the pH changes a ton because there's nothing there to interact with it. So as you add base, you are changing the pH of the solution. Then you hit the second pKa around 9.8, and again, you have to add in a lot of base to change the pH just a little bit. Does that make sense? Put a Q if it doesn't make sense. In this picture, we're showing that this is for alanine. If you had something, an amino acid like aspartic acid, where the side chain, the R group, also has something that can be associated and dissociated, it would have a third pKa. So in aspartic acid's case, the pKa of the side group is 3.9. And if we were going to draw this graph for aspartic acid at right around here at a 4, there would be another increase up where you would have to titrate in more base to change it. Then it would go over and have that other one again. Okay. So alanine can act as a buffer at 2 pHs, and aspartic acid could act as a buffer at 3. Okay. Okay. So in the blood, I just talked about amino acids, but what is the most prevalent buffer? It's actually bicarbonate. Okay. So I'm showing that here in blue. Bicarbonate can become carbonic acid, which happens quickly, and then that can become carbon dioxide and water. Now, the pKa of bicarbonate is 6.1, and our blood's around 7.35. So why is this our main buffer? It's not great by pKa. We would want a buffer that's around 7.3 for the blood, okay? But the reason we have this as a buffer is because we can exchange it with the outside world, right? If we need to, as we breathe out, we're breathing out carbon dioxide. And basically we have enough of it. So it's off by maybe, if it was 6.35, it would be off by 10-fold from what we'd want. But so it's off by maybe 15-fold or something. But it still works good enough. Sorry, and I think I just answered some of these questions. So, right, I said about 1 to 20, and it's good enough. Okay, so this brings me to another kind of thing I kind of want to work out, but I haven't done yet. Normally in class, when I have a poll everywhere or ask a question, I have you guys turn and talk to each other to try to figure it out, because it just makes your neurons fire and you guys learn better. I'd like to incorporate that some way in this class, and I'll have a word cloud at the end of today where you can throw out suggestions for how you'd want to do that. I could create groups for you. I thought you guys might want to have your groups and use Slack. You could use your anatomy groups. I started to play with grouping people by alphabetically by email. So I have a group set up for A and B and C and D emails, and you guys could video conference with each other. So we might try that later. But anyway, for right now, think by yourself, what would happen if you hyperventilate? Does that increase or decrease the pH? And then give me a C for comment. Okay, we got a bunch. Monica Schultz.
Wouldn't it increase the pH because diabetic ketoacidosis patients who small breathe, so they're breathing really fast to try to decrease their CO2 and increase their pH back up?
I didn't hear your rationale. I say that again. It is increasing the pH. Say why again?
I was just thinking DKA patients, who small breathe or they breathe really fast. They're basically tachypnic, and it's because they're trying to breathe off that CO2 and raise their pH back up to a normal level.
Right. So you have a real world example that it works. I'm trying to get at the mechanism of why that is. So if you hyperventilate, you're getting rid of carbon dioxide and you're actually increasing oxygen, right? So if you're lowering the amount of carbon dioxide, you're lowering this CO2. Draw this again, right? You're lowering this. From Le Chatelier's principle, you're going to drive in this direction, okay? So you're going to drive this whole thing over here. Okay, so you're going to lower the hydrogen, and that is going to raise the pH. Okay, very good. And then, does breathing in a brown bag cause an increase or decrease in pH? Keep things moving, I think you'd get that. That's going to do the opposite, right? If you breathe into a brown paper bag, you're rebreathing your carbon dioxide. So you're increasing the carbon dioxide here and driving the equation to the right and increasing the hydrogen. So that would lower the pH.
How about blue?
On my phone.
Why does it have to be brown?
Oh, sorry. All right. Okay. So we also, I just explained to you that bicarbonate is our main buffer, but we do use proteins a little bit to buffer and phosphate to buffer. Phosphate seems like a great buffer because its pKa is 6.8. It's much better than bicarbonate by pKa, but we don't actually have, we're using phosphates to modify proteins and stuff. And we don't have as much of it. It's not there in large quantities. So that's why it's not as big a player. Okay, so now back to our clinical case. A lot of this you haven't learned yet. We'll talk about type 1 diabetes versus type 2. Type 1 is where they can't create insulin. So the insulin is not produced, whereas type 2 is they're not detecting the insulin. In either case, you're not having the effect of insulin. And so in type 1 diabetics, you end up getting an increase in glucose in the blood and urine. And this is going to tie into next lecture. That causes an increase in water and solutes, so sodium and potassium in the urine. And it releases fatty acids. And then this is the spot that kind of was related to what we talked about. That ends up increasing ketone bodies, which have a low pKa. So because they have a low pKa, that lowers the pH of the blood. Okay. And so that's the detection of the acid. So here, again, you don't need to know any of this structures. This whole slide could be thought of as an FYI. I just wanted to explain to you kind of how you can think of words and what is actually going on. So here is an acid and an acid, right? A carboxylic acid and a carbonic acid. So they have the hydrogens to give up compared to its, this says conjugate base over here. Sorry, you can't quite see that. And then the relative pKa. So here's bicarbonate. It gives up its hydrogen to bicarbonate. Okay. And oh, I'm sorry. For a while, I'm repeating the poll everywheres in case we use Panopto, which is slower, so I can talk about them. So I'll be skipping over slides, but I want to keep that redundancy in case we need it. Okay. So given that slide I just gave you, do this real quick. I made up an acid. Schmugel acid has a pK of 4.5. Would it be more dissociated at pH 3.5 or 8.5? Ten seconds. It's interesting. The total results don't show, but then I think people can vote somehow. 5, 4, 3, 2, 1. Okay, 85%. And then people always change their answer. I gotta lock it. Okay, so right. So I told you it was an acid, right? So as an acid, it would have the hydrogen, right? So it has a hydrogen to give up. So at a pH, where would it be more dissociated? At a high pH, because there's not as much hydrogen around. At 4.5, it's half and half. So as you go up, its hydrogen is given up. Okay, at the end of every lecture, I have this poll, which is probably more important this year than any other year. So you answer it twice. Was the lecture, and then choose one of these 5, bad, below average, average, above average, and great. And then choose a second one. Was it way too slow, a little slow, just right, a little fast, or way too fast? So the best lectures would be great or above average and just right speed. So it'll help me kind of speed up or slow down. If I slow down, if I have to slow down a lot, then there might be assigned readings. I don't think I've ever been asked to speed up. All right. So with that, let's take a 10-minute break and we'll come back at 9:10. Okay.